Welcome everyone to our video on DNA replication. So before we dive into the details of this, let's just do a 30-second recap on DNA. So remember that DNA is a nucleic acid. It is made up of nucleotides, of which there are four: adenine, thymine, cytosine, and guanine. DNA is a double-stranded helix. It is composed of two strands of nucleotides. There's a sugar phosphate backbone, and then the bases project kind of outwards. One strand of nucleotides is going to hydrogen bond to the other strand of nucleotides, hence making it a double stranded molecule. These two strands are anti-parallel to each other. Notice how this top strand runs 5' to 3', and then the bottom strand is opposite, 3' to 5'. Not shown here are all of the bases that make up these strands, but we know that these two strands are complementary, meaning adenine will always pair with thymine and cytosine will always pair with guanine. And you will see more of that here momentarily. So, because DNA contains all of the essential genes needed for life, it's important that DNA be able to quickly and easily and efficiently replicate, right? Because before a cell can divide to make more cells, the DNA from that original cell has to be copied. That way, the newly made cell also has DNA. Okay, so this slide is just giving you a general overview of what's going to happen during DNA replication. And this function, being able to replicate and being able to code for proteins, has to do with the structure. So, because these two strands of nucleotides are held together by weak hydrogen bonds, the cell is able to break these hydrogen bonds of the parent molecule and separate them. Okay, so notice there's nothing happening to each strand. There's no changes being made. We've literally just think of it like almost like a zipper, like unzipping it. We're just detaching these two strands from each other. And then the cell is going to use each strand as a guide or a template. And it's going to be able to build a new second strand. And it can do this because the two strands are anti-parallel and complementary. By knowing the directionality and sequence of this top 5' to 3' strand, the cell can build from scratch a 3' to 5' strand. And then the same thing down here. So the cell is going to start with one parent DNA molecule. Okay, those hydrogen bonds keeping the strands together are going to break, allowing us to separate the two strands. Each one will be used as a guide to build a new second strand. And at the end of the event, there are going to be two daughter DNA molecules. Each molecule is going to have an original parent strand and a newly made strand. All right, so I'm going to go into presentation mode for the next slide. I have some animations here. So let's try to visualize this process. So here is the parent molecule, and I want you to notice that the two strands are anti-parallel. Guess we have our 5' end right here. Sorry, I should have just circled that. And the 3' end. So the second strand is opposite of that, 3' to 5'. And we'll see our base pairing, right? Where we have adenine on one strand, we have thymine on the other. If we have guanine on the top strand, we have cytosine on the other. Okay? So because of this, we are able to use each strand essentially as a guide. So let's check this out. So here's our starting parent molecule. The two strands are very close together due to hydrogen bonding between those bases. The cell is able to break those hydrogen bonds and separate those strands. Then the cell is essentially going to build from scratch new strands of nucleotides. So we'll start with the top strand. So because that top strand runs 5' to 3', the newly made strand is going to run 3' to 5'. So the cell is literally going to just read the top strand and build the bottom. So the first nucleotide in the top strand was an adenine. That means the newly created second strand needs to be complementary. That's with thymine. So then it just keeps going. The second nucleotide is guanine up top, so we need a cytosine down bottom. Then there's a cytosine at position number 3 on the top strand, so now we need guanine. And this just keeps happening, okay? The cell just adds one nucleotide after the other. And sorry, building this in PowerPoint means getting a perfectly straight line was just not happening. But then you'll notice at the end of this process, right, we now have a double-stranded DNA molecule. The two strands are anti-parallel and they are complementary. The same thing is happening with the bottom strand, but that template ran 3' to 5'. So the strand that it created is going to be anti-parallel. It ran 5' to 3', and it's complementary. And if you were to, superimpose these, you would see that they are the same, right? They've got the same directionality and the same sequence as each other. And so we say that DNA replication is a semi-conservative process. I'm sorry, I'm using white here. It is a semi-conservative process. And in biology, when we say something is conserved, that means it's the same. Okay. And so in this case, you think of semi as like, well, kind of, sort of, right? They're kind of, sort of the same. Do they have the same directionality? Yes, they have a 5' to 3' strand on top. Check. They've got a 3' to 5' strand on bottom. Check. If you compared the sequence of the tops and bottom strands, you'd see that they were the same. So that's where the conservative part comes in. The semi means that one strand in each of these molecules, let me try to ditch the red here. I'm sorry, ditch the yellow. One strand in each molecule came from the original parent strand. Each strand, each new DNA molecule also has one newly created strand. So that's what we mean by semi-conservative. Okay, both strands in the DNA molecule are not of the same origin. One originated from the parent molecule and one was newly created. All right, and so remember, we do call these two DNA molecules that are present at the end of the process the daughter molecules. All right, let's real quick zoom in on DNA replication. So DNA replication begins at sequences called the origin of replication. And this is where those parent strands are initially going to separate. Now, bacteria only have a single origin of replication on their DNA. Eukaryotic organisms have several. So I'm going to show you at the bottom what's happening in a eukaryote only because it's really going to drive home the process of separating the strand of DNA and things like that. So at the origin of replication, which I have indicated by these dashed lines, this is where the DNA starts to separate. And when it separates, we call this a replication bubble. Within that bubble, DNA is going to be copied bidirectionally. Okay, bidirectionally. And that's what you see here. The DNA is going to be copied going to the left and to the right. That's happening both on the top strand and the bottom strand. Because remember, this has to be a really quick and efficient process. If we think about bacteria, even though they only have a single origin of replication, they divide completely from one cell into two, normally in about 20 to 40 minutes. So that means the DNA has to be replicated in a much shorter time than that. So that's really quick. Okay. So once the DNA in the replication bubble has been copied, the bubble expands. The DNA replication process continues until eventually the whole thing has been copied. Okay. And we end up with those two DNA molecules. All right. So here we have daughter molecule number 1 and daughter molecule number 2. Again, it's semi-conservative because each daughter molecule of DNA has one original parent strand and one newly made parent strand, as indicated here. All right, so let's go ahead and wrap up this video with an apply what you know. So a parent DNA molecule is shown below. Sketch the two daughter molecules that will be made after DNA replication. Okay, label the parent strands and the newly made strands. So remember, essentially what's going to happen here is that this parent DNA molecule is going to be separated. They're going to break the hydrogen bonds that exist between the strands. And so I'm just going to write dot, dot, dot here. So these strands will be separated. Okay, and then each one is going to be used as a template. So I'm actually going to try to get blue here to kind of match the starting color. So, all right, so my parent strand is going to be GCATT, GCAAG 3 prime. And then my 3 prime strand is CGTAACGTTC 5 prime. Okay, now I do recommend you pause it here and you try to build out the other sequences and strands from here. And I'm going to go ahead and switch colors back to red. Okay, so remember each of these strands is going to be used as a template. So we know if I'm working on this top strand first, the parent strand runs 5 prime to 3 prime. So I know the newly made strand is going to be opposite because they're anti-parallel. And because they're complementary, I can use base pairing to figure out what the sequence will be. If there's a G in the parent strand, the newly made strand will be a C. If I have a C up top, it's going to pair with a G. A pairs with T. T pairs with A, G with C, C with G, A with T, A with T, G with C. Now, that's daughter molecule number 1. And again, if you check the directionality as sequence of the original bottom parent strand, you'll notice it's the same because we need the daughter cells to be the same as the parent strand in terms of sequence and directionality. So now I'm going to do the bottom strand. The parent strand runs 3 prime to 5 prime. So the newly made strand will be 5 prime to 3 prime. And again, I'm going to just use base pairing to figure this out. So there we have it. All right, y'all. Actually, I think there's one more. So here's an explain it. Okay, and I'm just going to talk through this one. I'm not going to write anything on the slide. This is just to give you exposure to what some open-ended questions may look like on assessments. So if a student says the cell has to keep both original DNA strands together because they already contain the correct genetic information. So if a student says that, what's your response going to be? Can you explain why this statement is incorrect? Use the concept of complementary base pairing in your explanation. So we know that the strands can't stay together, right? We need to be able to copy this DNA molecule, and that's just not going to happen if the strands remain together. And so the cell is going to separate those original strands, the parent strands, by separating the hydrogen bonds. It's going to use each one as a template to build a new second strand. So with what this student is describing, that would be called a conservative model of DNA replication. If somehow, doesn't happen, but if somehow the DNA was able to copy the 2 strands and then bring the 2 parent strands back together, then that would be a more conservative model. But research has shown that that's not actually what happens. That is what people originally thought. But we now know that um, they just separate those original strands, use them as a template to build new strands, right? Because if you think about it, DNA is so critical, right? We don't want to separate the parent strands, use them to build new strands, and then try to separate those just so the original parent strands can go together and so the newly made strands can go together, right? You're making it really risky, right? Because a lot can happen inside the cell that could then destroy or mess up or make vulnerable those DNA strands. So you want to do as little separating and rearranging as possible. Okay, so that's going to be why once those parent strands separate, they're staying apart. They're just going to build a new strand and keep those molecules intact. All right, y'all. That is going to be it for this video. Please let me know if you have any questions. Bye, y'all.